Okay, this is a tutorial video on measuring the fluorescence amount, the amount of fluorescence in our cleaning samples. We're going to start with just a couple of example images, and this is how you set it up. So if you're going to do a new color, this is one way to figure that out. So you open ImageJ. Here's the ImageJ window here, and we're going to just start with the dirty image because then you have plenty of fluorescence to analyze. You can drag and drop it onto this window, and there's our image. You can see 3 capillaries. They have varying amounts of soil in there. This is spin 22, a cutting fluid that has fluorescent property built in. I don't know if they added a dye when they made it, but it definitely fluoresces. So what we want to do first is we want to adjust this image, and we're trying to make it into a black and white image where the black pixels are the fluorescence and the white pixels are everything in the background. So we're going to do a color threshold. So under image, adjust, color threshold. Now there's different ways to operate. In the past, we always have been using RGB values, but I think HSB is better. So if we do RGB, we see that there are some pixels in green and red and blue. And I tried to do this with RGB, and it didn't work as well. So we're going to use the hue, saturation, and brightness scale, because this allows us to separate all of the colors, sort of a recombination of the R, G, and B values. And so what we want to do is we want to just open up the brightness all the way, open up the saturation all the way, and then we're going to filter on the color only. So you can look at this. As I move this up, I start to see the fluorescence kick in. Okay. Now we want the fluorescence to be black and the background to be white. Okay. So we're going to come down here. We don't want those are the fluorescing pixels. So we want to make sure we don't cancel those out. We want to cancel out the background. So if we come down here, you see all of those blue pixels from the fluorescing background are now eliminated, and only the fluorescence color is captured. So here we're picking that kind of blue. So we're just wanting that fluorescent, that shade of blue in the original picture. You can see the original picture here. Okay. And this is the filter. So that's a real nice thing. You can look at the original. You can see we want just those light blue pixels, which have this color right here, this hue that we're capturing. Okay. And so we want that hue to be black and everything else to be white. Okay. So I've analyzed this, and about 120 is good, and then up here 160. So we're going to go with 160, and that's a very consistent measurement of our fluorescing color for spin 22. Now, we've made this a black and white image, so it's the active image. So then we can come up here and do analyze, and we're going to count particles. Now, it's sort of treating this as particles, but we really just want the total number of black pixels in this image. So that would be on the summary. So we're going to do analyze particles, and then we're going to see this summarize is what we want. So we're going to hit OK. And there's the total area of black pixels in this image. Okay. The nice thing about this is we can leave this dialog box open, and we can leave our threshold color the way it is, and we're going to bring in the cleaned image. So don't change anything, just drag the next image in this set. Here's the cleaned image, and you can see there's still a little soil left over from the cleaned image. And we come to the threshold color, and notice it changed this brightness value, but when we make this go back down to 0, or change it at all, it applies the filter to that image. We still have the 120 and 160 for our hue. We still have 0 to 255 for saturation, 0 to 255 for brightness. And we now have the cleaned image with its residual soil there. So we come back up here to analyze particles. Summary, hit OK. Look, it adds it right there in the same dialog box. So we can analyze our dirty and cleaned image here. It will keep those file names right here, and it'll give the total area of soil in each file name. So this is a fantastic result. So if we have all of our images properly labeled and we analyze them this way, we'll get these little tables, and we can come up here and we can save as, and it goes hopefully into the default directory, but you can search around till you find it. And we can call this whatever the image name is. Like if we click on this dirty image, and we can change this, instead of a JPEG, we want a text file, TXT. So I hit save, and now it saves this little text file that has our data in it. And then we can put that in Excel and we can analyze these things. So this is a fantastically simple way to analyze the fluorescence. The only trick is to find the correct hue. So when we have a different dye that may be a little more green or a little more yellow, we're going to have to move this little envelope of color so that we get the fluorescent color and eliminate all the other colors. That will allow us to define the pixels as soil or background, and then we can use the analyze particles to count up the number of pixels of soil. So hopefully that's clear, straightforward, simple, and the data output is really nice and convenient. We can put those into Excel very easily.